Hey everybody, in this video I want to talk a little bit more about RNA interference. So RNAi, and I already talked a little bit about how RNAi was discovered. In the previous video I covered that. In this video I want to talk more about how double-stranded RNA specifically induces RNA interference. Also how microRNAs, what are microRNAs and how they can also cause RNA interference. And then I want to talk about how small hairpin RNA encoding genes are used in research. So essentially, what is RNAi? Just a brief review. RNAi is when RNA molecules are used to prevent translation of a, let's say, complementary messenger RNA. What is this thing here? Let me erase that. Okay. So, this is a very general definition of RNA interference. A lot of researchers will say specifically when double-stranded RNA molecules are used to prevent translation of a complementary messenger RNA. But microRNAs can also cause RNAi. And whether or not they're double-stranded RNA is a matter of semantics. So we're just going to use the general definition as RNA molecules used to prevent translation of a complementary messenger RNA. So now let's cover the mechanism by which this occurs. So let's say a double-stranded RNA molecule somehow finds itself in a mammalian cell. And this double-stranded RNA could have been placed in the cell by a researcher. Also could be a result of a virus that possesses a double-stranded RNA molecule as part of its life cycle, that forms a double-stranded RNA molecule as part of its life cycle. So this double-stranded RNA should be longer than at least 25 base pairs to trigger RNAi. And we're going to see why it should be longer than 25 base pairs in a moment. So, double-stranded RNA, what does that mean? Well, essentially it's made of two single-stranded RNAs that are complementary to one another. Now, if this molecule should find itself in a cell, a mammalian cell or a plant cell or a fungal cell, all of these different eukaryotic cell types will process this double-stranded RNA in the same way. So an enzyme called dsR, so this is a protein, an enzyme called dsR will chop up that double-stranded RNA into small or short interfering RNAs. Now these short interfering RNAs, depending on which dsR, some organisms have multiple dsRs, but depending on which dsR chops it, will be between 21 and 24 nucleotides, or let's say base pairs long. And they are called short interfering RNAs. And let's say that was a long double-stranded RNA molecule up there. So we can get lots of these short interfering RNAs produced from that double-stranded RNA when it is processed by dsR. And so each of these are, let's say these are all 21 base pairs long. Now, dicer processes double-stranded RNAs in such a way that most of the molecule is double-stranded, but we have these two nucleotide overhangs at the 3' ends. So that would be a 3' end here, 3' end there, 5' end there, 5' end there. Okay, now each of these is a short interfering RNA, or siRNA. Sometimes people just call them siRNAs. Now these are used by another enzyme, actually an enzyme complex called the RISC complex. RISC stands for RNA-induced silencing complex. So in a very important protein, probably the most important protein of the RISC complex, is called argonaute. So the argonaute protein is a component of the RISC complex, and it binds to the short interfering RNA. Actually, it binds to one strand. It uses only one of the strands, and the other strand is lost. There's a complicated mechanism by which one of the strands of the short interfering RNA is loaded into the RISC complex, but we won't discuss that. So we just need to know that the RISC complex with the argonaute will bind one of those strands from the short interfering RNA, and it will use that as a guide to screen screen the messenger RNAs in the cell for complementarity. And if it finds a messenger RNA with a complementary sequence, it will cleave that messenger RNA. So let's say this is our messenger RNA with a 7-methylguanosine cap and a poly-A tail. And let's say the short interfering RNA that has been incorporated into the RISC complex, let's say it matches right there. It's complementary. And remember, this is about 21 nucleotides long. So if it matches, if it's let's say a perfect match, what will happen is the argonaute protein within the RISC complex will cleave. Maybe I can draw some scissors here. Here are my scissors. It will cleave right across from about the middle of that short interfering RNA. And when that happens, it can't be translated, right? That messenger RNA can't be translated anymore by functional proteins. Plus, we've got these free ends here that can be degraded by exonucleases. Exonuclease. So that messenger RNA can be degraded. So, and that would be a good thing, right? If this double-stranded RNA was a virus, well, you can see the cell has a really neat way to identify messenger RNAs from that virus. So let's say this is a virus messenger RNA that would encode a viral protein that is needed for that virus to replicate in the cell. Well, the cell has a really neat way to identify those viral messenger RNAs and prevent them from being translated. And you might be asking, how? Well, if cells possess this process, how come we have viruses still that possess a double-stranded RNA molecule as part of their life cycle. Well, these double-stranded RNA viruses, they've evolved mechanisms to bypass or to evade the RNA interference machinery. It doesn't always work, but some of them have methods to evade RNAi. But the presence of RNAi makes it harder for them to infect eukaryotic cells. So now researchers can use RNA interference by injecting cells with double-stranded RNA. It's a little complicated with mammalian cells because there's another, I think, antiviral defense process that is activated when long double-stranded RNAs are put into mammalian cells. But other types of eukaryotes, well, scientists can just inject double-stranded RNAs into those cells or get double-stranded RNAs into those cells, long double-stranded RNAs, and genes can be silenced through this mechanism that I just discussed. So for mammalian cells, researchers have come up with another method for using double-stranded RNA-based RNA interference to silence genes, and that involves short hairpin RNA encoding genes. I'm going to talk about this towards the end of this video, but what these do is they bypass the need to introduce long double-stranded RNAs into a mammalian cell, and by introducing a short hairpin RNA, well, RNA interference can be activated in the cell without activating other processes that would interfere with research. But we're going to talk more about this towards the end of the video. Okay, now that we know how double-stranded RNA triggered RNA interference works, we can talk about how microRNAs trigger RNAi. So microRNAs. Now, what are these things? Well, I'll just start by saying, so it's thought that humans have around 2,000 microRNA encoding genes. And these are really interesting, and we haven't talked much about these. We spend so much time trying to, understand how protein coding genes are transcribed and turned over and translated that we don't really have a whole lot of time to talk about all these other interesting types of genes that exist in humans or other organisms for that matter. So in humans, there are about 2,000 of these things. So these are transcribed by, in general, they are transcribed by RNA polymerase II, just like protein encoding genes. And they are capped. I think it's a 7-methylguanosine cap. I'm not really sure, but they are capped. I imagine it's a 7-methylguanosine cap because it is RNA polymerase II. And they are poly-A-tailed. So, as an example, let's assume this diagram right here is of a microRNA encoding gene. Now, here is, let's say this is the promoter where RNA polymerase II would bind. This is the +1 site. And so let's say this is the transcript. It's been capped. There is a poly-A tail. Now this is going to fold. Why is it going to fold? Because of the sequence of the microRNA gene. This is going to fold, and it's going to form a structure that looks something like this. I want to redo this a little bit. So we're going to get this loop right here and this hairpin structure formed. And this forms because this sequence right here is going to be complementary to that sequence down here. and it's usually not perfectly complementary. But it's complementary enough to get a stable hairpin to form. And this is called a primary microRNA. So these primary microRNA, so if we went back and looked at this gene up here, we would see sequences that are very similar but inverted with respect to one another. So let's say that sequence here would be this end over here, and this sequence here would be this end over here. So the primary microRNA, often abbreviated as a microRNA, is going to be processed by an enzyme called DROSHA into a about 70 nucleotide long RNA molecule that forms this hairpin. So essentially, you can see what happened by comparing this diagram down here with this one up here. We've lost all this stuff. That's all been removed. Now this is going to be called the pre-microRNA. Now this step right here is thought to occur in the nucleus. And this is the pre-microRNA is exported to the cytoplasm, and that's where dicer is. And when it's in the cytoplasm, dicer, just like dicer does to long double-stranded RNAs, dicer will process this pre-microRNA into something that looks a lot like an siRNA. So these little bumps right here, this is indicating that this is usually not perfectly complementary. So the base over here isn't complementary with that base over here. Now this structure right here, this is called the microRNA. I'm just going to call this microRNA star complex. So only the microRNA, only one of these strands is the microRNA. The other one is the microRNA star. Only the microRNA, well, usually, most of the time, only the microRNA strand, the microRNA, is incorporated into the RISC complex. So just like the siRNAs, one strand of the siRNA is incorporated into the RISC complex, only one of these strands is going to be incorporated into the RISC complex, and these microRNAs have evolved, so it's usually the microRNA, not the microRNA star, that's incorporated. Now, the microRNA is going to be also called the guide strand. In the microRNA star, we are going to call that the passenger. And there's a special complex that will load the microRNA into the RISC complex. So lots of different complexes involved. But in the end, what we get is that RISC complex with an Argonaut protein, Argonaut, will incorporate that microRNA which is about 22 nucleotides long. So for microRNAs and short interfering RNAs, we can always assume between 21 and 24 nucleotides long, depending on which dicer is doing the processing. It might be 21 or it might be 23. So just remember between 21 and 24 for the length of these things. And okay, so what will happen now? Well, at this point, it's essentially just like the other pathway that we talked about, which is we have our messenger RNA in the cell, and the RISC complex can scan the messenger RNAs. And if it matches the microRNA, well, translation of that messenger RNA can be suppressed. Now, microRNAs are interesting in that they very often do not match the targets perfectly. So these microRNAs, they exist to regulate genes in the same genome, right? To regulate protein coding genes. So we have microRNA encoding genes which are expressed to prevent translation of complementary messenger RNAs from genes in the same genome. So it's another way for the cell to control which genes are, or which messenger RNAs, I should say, are translated into proteins. So it's another method of gene regulation. So in addition to controlling, if a gene is transcribed into a messenger RNA, once that messenger RNA is in the cell, well, microRNAs can be used to determine if that messenger RNA can be translated. So something interesting about microRNAs, they often do not match perfectly to the target messenger RNAs. There is what we're going to call the seed region, where we have to have like a near perfect match. And for the rest of the microRNA, it doesn't have to match that well for that microRNA to still suppress translation from the messenger RNA. We're going to learn more about the seed region and microRNAs in the next lecture. So one other thing I want to touch on here. So there are two mechanisms by which translation can be suppressed. So one is that messenger RNA can be cleaved. And it's widely thought that if the siRNA or the microRNA matches the messenger RNA perfectly, then the messenger RNA will be cleaved. So we can also just have translational suppression. So just having that RISC complex stuck on that messenger RNA, translation of that messenger RNA can be suppressed by mechanisms that aren't always quite that clear yet. So translational suppression is thought to result when we have an imperfect match. So the seed region matches, but the rest of that short interfering RNA or microRNA doesn't match so well to the messenger RNA. Well, then we'll get translational suppression, but not cleavage of the messenger RNA. But overall, two different ways the translation of the messenger RNA can be prevented: by destroying the messenger RNA or just preventing translation without destroying the messenger RNA. So, okay, now that we covered the mechanism of RNAi and with respect to RNAi triggered by double-stranded RNAs or microRNAs, we can talk a little bit about how researchers use short hairpin RNA encoding genes to silence genes for research purposes. So, and the reason I want to talk about this, in an upcoming lecture, we're going to see a research article on where researchers use short hairpin RNA encoding genes as part of, in the article, or in the research they are performing. So, with mammalian cells, what researchers usually do is use a lentiviral vector. And another reason why I want to talk about these is we've already learned a lot about using lentiviral vectors in gene therapy, right? And so remember, a vector essentially is any vehicle, a virus or a plasmid, that is used to ferry a DNA sequence into a host cell. So, and with lentiviral vectors, remember when we're making these lentiviral vectors, we usually need 4 different plasmids. So this is just a review right now, because if you're anything like me, you probably forgot, how these lentiviral vectors work, even though, it's been a few weeks, so we need a little review here. So there's the packaging plasmid, there's the envelope plasmid, there is the regulatory plasmid, and there is the transfer plasmid. So we use these 4 plasmids, and we introduce these 4 plasmids into a packaging cell, so some mammalian cell line. And what do these 4 plasmids do? Well, the packaging plasmid has the GAG gene, which essentially has the capsid protein and the nucleocapsid protein, and the POL gene, which has that reverse transcriptase, the protease, and the integrase, and hopefully some of this is coming back to you as I describe it. So the envelope plasmid, well, this is needed for fusion of the virus, the lentiviral vector, for fusion of that with the target cell. So, and if you remember, what did HIV use? I think it was GP41 and GP120. And it doesn't matter. Well, the protein we use in our lentiviral vector, it might be those. Often it's one called, I think, VSV, because a VSV protein is great for getting your virus into a broad range of mammalian cell types. But anyway, the envelope plasmid is going to have that envelope protein that we need for the virus to fuse with our target cells. The regulatory plasmid has a gene called REV, which is needed to transport some of these viral RNAs that are going to come from this plasmid in the packaging cell. And the transfer plasmid is going to have the gene in between the LTRs, right? The long terminal repeats that are typical of a lentiviral genome. So the gene we want to put in the cell is going to be between the LTRs in the transfer plasmid. So all 3 of these plasmids, they're all set. We don't need to do anything there. They're the same every time, essentially. We may change out the envelope plasmid for a different one now and then, depending on the target cell. But this is the one we need to modify if we want to use a short hairpin RNA encoding gene to silence a gene in a target cell. So essentially, okay, so the question is, how do we silence a gene? Let's say gene X, any gene, we could put any gene there, with RNAi using a lentiviral vector. And lentiviral vectors, these are used with mammalian research. If you're working with a mammalian cell line, you're going to use a lentiviral vector to get your gene into the mammalian genome. So essentially what we need to do is just modify that transfer plasmid. How are we going to modify it? We're going to modify the transfer plasmid to carry, we want it to carry the short hairpin RNA gene between the LTRs, right? And so let's say the plasmid, here's a diagram of the plasmid with the one of the repeats here. Now maybe you remember these repeats are not full length with the lentiviral vector, but they look something like this. Remember they had sequences that were, well, we had a U3, R, and U5, and the one on the left here, 5' end, was incomplete. So, and here we're going to have an RRE element, which is an extra detail we probably don't need at this point. But anyways, that, remember how we had that Rev plasmid? So the Rev protein binds the RREs and helps transport these sequences in RNA form throughout the packaging cell. So, okay, now this is where we would have that short hairpin RNA encoding gene. So right in here. And okay, what would that look like? So essentially what we would need is a promoter and a 21 base pair sequence that matches our target gene. So what is the target gene? The target gene was gene X. So we need a 21 base pair sequence from that gene. It should be found in an exon of that gene too, not an intron. And I'm going to put a little arrow right there for now. Then we're going to need a 6 base pair, about 6 base pairs, random sequence. Could be anything we want. Then next, we're going to have this same 21 base pairs right here from gene X inverted after that 6 base pair random sequence. And then we're going to end with a terminator sequence. Now, typically the promoters, we haven't talked much about these types of promoters, but typically it's an RNA polymerase 3 promoter and an RNA polymerase 3 terminator. RNA polymerase 3 promoter. And that's it. And so what will happen is that when this is transferred into our target cell, okay, let's say this is in the transfer plasmid. So we produce the lentiviral vector. We put all 4 of those plasmids into the packaging cell, and what comes out are the viral particles with the fusion protein on the surface and the viral genome here is going to have our short hairpin RNA gene in there, and no viral proteins, right? Just a short hairpin RNA gene between those partial LTRs. And if this is our target cell, this is the target cell line. This is the target cell line that we want to reduce or silence the expression of gene X in. So we mix the viral particles, the lentiviral vector particles, with the target cell. And then in that cell, the short hairpin RNA encoding gene will be incorporated into the genome. And well, what happens? So essentially, when this is transcribed in the cell, we get something that looks like this. A short hairpin RNA with a 6 base pair loop. Now this, right, so transcription of the short hairpin RNA encoding gene, because these sequences here are inverted relative to one another, that RNA, which is going to be what? It's going to be 21, 42, about 48 base pairs long, is going to fold because of intramolecular base pairing, fold into a hairpin structure like this. Dicer, which is in all mammalian cells, is going to chop that into a small interfering RNA that can be incorporated into the RISC complex. And what gene or what messenger RNA is going to be complementary to this small interfering RNA from the short hairpin RNA? Gene X, right? So matches or is complementary to gene X. So this RISC complex now is going to search the cell for messenger RNAs from gene X, and it's going to cleave those because this is going to be perfectly complementary to gene X. So this cell line is no longer going to be able to express normal levels of gene X, of the whatever protein is encoded by gene X. So it's going to produce significantly less of that protein. So one of the nice things, if you work with mammalian cells, is there is a group called the RNAi Consortium, so TRC, that has already made essentially lentiviruses or lentiviral particles that you can order for essentially any gene in the human genome or any gene in the mouse genome. And so you don't even need to make the viral particles. You can just order them. You have to purchase them, but you can order them, and you can mix them with your cell line of interest and see what happens when your cell can't express that target gene. So, okay, yeah, so that's it. So we're going to see a little bit about how researchers use short hairpin RNAs to study a special type of RNA molecule that has only really been, it was discovered a while ago, but only recently have they been, I guess, viewed as important and not just junk RNAs. So these are called circular RNAs. And one of the primary functions of a circular RNA, one of the main functions is thought to be to act as a microRNA sponge. And we'll talk more about what that means in the coming lectures. Okay, I hope you enjoyed the video. I hope it helped you understand a little bit about how microRNAs work, how short interfering RNAs work, and how short hairpin RNA encoding genes can be used to silence genes in research. Please, if you have any questions, come to the Zoom Q&As or send me an email. Okay, see you in the next video.